Good day. This is the Professor, CBMD PhD. It's a Sunday, May 28, 2023. It is 6:13 and 29 seconds, p.m. Eastern Daylight Time. And today's research, infrared lasers. So let's jump to it here. New study shows how infrared lasers destroy harmful protein aggregates in Alzheimer's. Date August 4, 2020. Source: Tokyo University of Science. The agglomeration of proteins in the structures, called amyloid plaques, is a common feature of many degenerative diseases, including Alzheimer's. Now scientists review, through experiments and simulations, how resonance with an infrared laser, when it is tuned to a specific frequency, causes amyloid fibrils to disintegrate from the inside out. As their findings open doors to therapeutic possibilities for amyloid plaque-related neurodegenerative diseases that have thus far been incurable. In recent years, instead of going down the chemical route using drugs, some scientists have turned to alternative approaches, such as ultrasound, to destroy amyloid fibrils and halt the progression of Alzheimer's disease. As scientists present the results of laser experiments and molecular dynamic simulations, this two-pronged attack on the problem was necessary because of the inherent limitations of each approach. While laser experiments, coupled with various microscopy methods, can provide information about the morphology and structural evolution of amyloid fibrils after laser irradiation, these experiments have limited spatial and temporal resolutions, thus preventing a full understanding of the underlying molecular mechanisms. On the other hand, though the information can be obtained from molecular simulations, the laser intensity and irradiation time used in simulations are very different from those used in actual experiments. It is therefore important to determine whether the process of laser-induced fibril dissociation obtained through experiments and simulations is similar. The scientists use a portion of a yeast protein that is known to form amyloid fibrils on its own in their laser experiments. They tune to the frequency of an infrared laser beam to that of an amyloid band of the fibril, creating resonance. As scanning electron microscopy images confirm that the amyloid fibrils disassembled upon laser irradiation at the resonance frequency, and the combination of spectroscopy techniques reveal details about the final structure after fibril dissociation. As for the simulations, the researchers employed a technique that a few members of the current team had previously developed, called non-equilibrium molecular dynamics simulations. Its results corroborated with those of the experiment and initially clarified the entire amyloid dissociation process, down to very specific details. As through the simulations, the scientists observed that the process begins at the core of the fibril where the resonance breaks intermolecular hydrogen bonds and thus separates the proteins in the aggregate. The disruption to this structure then spreads outward to the extremities of the fibril. And together the experiment and simulation make a good case for a treatment possibility for neurodegenerative disorders. In view of the inability of existing drugs to slow or reverse the cognitive impairment in Alzheimer's disease, developing non-pharmaceutical approaches is very desirable. The ability to use infrared lasers to disassociate amyloid fibrils opens up a promising approach. The team's long-term goal is to establish a framework combining laser experiments with non-equilibrium molecular dynamic simulations to study the process of fibril dissociation in even more detail. And new works are already underway. All these efforts will hopefully light a beacon of hope for those dealing with Alzheimer's or other neurodegenerative diseases. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.